Yesterday, we learned about guide RNAs and Cas9 components. Like Cas9 components have two different domains, which larger lobe and then smaller lobe. Larger lobe, which contains REC1, REC3 domains for the recognition of the target genes, followed by the smaller domain, which consists NUC, HNH, and PAM interacting site, followed by the recognition domain number 2. These two domains were linked together, called as in beta bridge beta helix, the domain it's like a small amino acid peptide which links together. This is the Cas9 component. When the Cas9 component were attached towards the target sequence, thereby the recognition of the PAM site, then only the Cas9 protein, the complex, were activated in the for the efficient knockout or any knock-in process. For this, uh, the guide RNA, which contains the target with the complementary base pairs that end with the NGG. This is called as a guide RNA. For the efficient knockout or efficient knock-in, we need to design the guide RNAs. For the suitable, we need to design the suitable guide RNAs for the efficient knockout process. For this, we need to check with the various analysis like sequence retrieval systems, and followed by the efficient guide RNAs designing with various software tools, and then to check the specificity and then efficiency analysis. This three major steps for the designing of the guide RNA. After that, we need to check with the efficiency. Then we'll directly go with the synthesis process from the various reputed companies like Europeans, and several other companies were available in Bangalore, Chennai, Hyderabad, Delhi, Bombay. For this, we need to get the guide RNAs. Then we can use for our respective experiments. First, we'll discuss about the like target gene identification and sequence retrieval system. First, all the sequence and then all the sequence databases were stored in the particular basic platform that is called as an NCBI GenBank, National Center for Biotechnology Information GenBank. This is a website for to store the n number of species from the larger in size or the smaller in size, whatever it may be. This is the basic site to store the respective genome of our interest, not only genome, transcriptome, all the primary databases were stored in this particular database. Apart from NCBI, we have plenty of softwares like Ensemble alone, and then Ensemble plants, Phytogome for efficiently used for the plant sequence retrieval system. Apart from these, n number of individual platforms, like in the case of Arabidopsis, in the case of plants means Arabidopsis thaliana. For Arabidopsis thaliana, we have peculiar database like Arabidopsis thaliana Resource Center, that is called as an ABRC, Arabidopsis Biological Resource Center, which is hosted by the Ohio University in US. Along with this, a number of individual platforms like soybean have a specific platforms called as an GLIMA sequences. GLIMA databases, which is also offered by the GLIMA Institute, which is located in US, along with in India, one of the premier institutes like University of Hyderabad were hosted the tomato whole genome sequencing and then tomato whole genome platform for the peculiarly for the Lycopersicum esculendum sequence retrieval systems. For this, we can retrieve the sequence for the efficient gene knockout or the knock-in process. First, yeah, this is NCBI. I think all the peoples have a hi-fi network facilities. In the case of, you can browse the platforms using have provided web links, web link also. You can you can type and then check with the respective site also. This is NCBI GenBank homepage. Like several can analyze the blast, we can compare with the various sequencing along with the genome sequence, SNPs, and the gene, protein, PubChem, whatever it may be, we can retrieve the sequences from the primary database called as an NCBI. Yes, we can, shall we start the sessions like in practicals hands-on training? First one, whether my screen is visible for the participants.
Yes, so it's visible.
Currently, I'm the visible,
Yes, sir.
Yes, sir.
not in a PPT. I am working in
Yes, sir.
databases like NCBI, like that.
No, sir.
No?
The PPT is showing.
Okay, okay. I will share my entire screen. You can check with that also. Yes. I will stop the sharing of my PPT, followed by I will share the entire screen. Yes. Now the screens are visible.
Yes, sir. We
Yes,
can
sir.
see the
Yes,
NCB.
sir.
Yes. Yes. Thank you. Thank you. Thank you. Yeah, this is the homepage of NCBI. This homepage will look like this. Here we can click the all databases, and then we can select the nucleotide option. Here we can select the nucleotide. If you want to select the all databases, we can directly go to that also. I will type the MYB12. This is MYB12 is an peculiar transcription factor for the efficient secondary metabolite productions in uh, plant species. Like this particular transcription factor will overexpress, means the entire phenylpropanoid biosynthetic pathway genes were upregulated. Finally, the end product is called as an flavonols. Flavonols means quercetin, rutin, camphorol, myrcetin, fistin. These and all some peculiar flavonols for the overexpression, means we can induces MYB12 transcription factors. This is the basic information about MYB12, and then directly we can go to the search option. Then, yeah, the next home page will looks like this. From this, we can choose the nucleotide option, or otherwise, first I have showed that the box which we can click the nucleotide itself. Yes, we can reduce the search options, means we can check with the plants alone, or otherwise we can click with the plants. Yes, from this around plant around 498 nucleotide sequence were submitted in various plant species. Apart from this, I have chosen the Arabidopsis thaliana MYB12 transcription factor. Like I need to click the Arabidopsis thaliana from the results by taxon clarifications. After this, yes, we need to choose the Arabidopsis thaliana MYB12 MYB domain protein number 12 mRNA sequence. Yes, we can click this option, and then yes, this is the complete GenBank sequencing, GenBank submission informations. From this GenBank information, we can get the complete genomic informations, like from the accession number, and then which the position of the chromosome were, the particular gene were located in the chromosomes, like the locus, followed by the how much base pairs in size, whether that is an mRNA or rRNA, tRNA, and then complete genomic DNA, along with the whether this is a linear sequence or circular sequence or supercoiled sequence, followed by the submission date also, we can check that. And then also like source of the DNA materials like Arabidopsis thaliana, and then complete taxonomic informations like from the top to bottom, from the class to species, until we can get the informations like along with the authors in for the submission authors, along and then followed by the title of the sequencing information, followed by the journal informations like journal paper title, journal title, and then PubMed ID, along with the here we can also we can check with the sequencing information from the source 1 to 1583 base pairs. From this, the gene sequence, we can click this, means this is the complete gene of the CDS, the complete coding sequence from the MYB12 transcription factor. Yes, this is the complete information about the like what GenBank information of the Arabidopsis thaliana MYB12 transcription factor. We can check the sequence in the format of FASTA. From this, we can click here, or otherwise we can click the FASTA format, fast alignment search tools. We can retrieve the sequence in the format of FASTA. We can copy the sequence and paste into new notepad. Like we can check the notepad. Yes, we can paste into new notepad file format. Yes, this is how we need to access the NCBI's platform to retrieve the sequencing information from the previously submitted data. And this is one of the option, followed by the second one. Second one is ensembled. Ensemble plants. This is also one of the premier easily available, freely available sequence retrieval platforms were combined together with the NCBI major platform also to link together with same. From this, we can click with this platform is peculiarly for plants alone, or otherwise if you want to check with the microbes, animals, and some other kind of rodents, humans, means we can directly go with the ensemble platform alone. For this platform, peculiar for the plant species alone. From this, we can check with the Arabidopsis thaliana, and for have checked with the some of the genes like MYB100. This is a kind of transcription factor. This transcription factor is negative regulated for the flavonoid, isoflavonoid biosynthetic pathway in plants. Okay, this transcription factor is there, means the complete isoflavones. Isoflavones means genistein, diastein, glycitin, and then this kind of secondary metabolite production were suppressed. What is the major use of isoflavones? Isoflavones are the trademark for the n number of antioxidant, n number of clinical properties have, but isoflavone, the particular compound was highly presented in the soybean. Okay, that's why from this search I have checked with the glycine max. Glycine max, MYB100 transcription factor. We need to retrieve the sequence. That's why have select with the glycine max, and also have mentioned like MYB100. Then we can go with the sequence. Yes. MYB 100. Sorry, this gene was not presented in ensemble plants. We can new search and then directly go with the glycine marks. Similar like MYB 12, MYB 12. You can directly check with this. Yes, this is MYB 12. I've showed in two submissions. We'll check with the first submission, like first one, glyma sequence of the particular sequence. Yes. Yeah, we can. This is the complete coding sequence of the MYB 12, which is present in the glycine marks. Yes, we can see that this is the sequence where the coding sequence, like we can check with the first protein ID and then amino acid length, base pairs, positive strand or the reverse strand, then followed by the coding sequence or the non-coding sequence, and then location, in which position the particular gene was presented. All the informations were denoted by this particular software. Like we can go with the gene sequence. Yes. Yeah, transcript ID. Yeah. This location of the chromosome was 6, and then position, reverse strand, and then transcript. This complete transcript of one exonic region, and then 18 domains, and then further features, and then 29 oligoprobes, along with the transcription gene of the transcription, the ID. We can click with the first exonic region. We can displace the sequence like this. The complete sequence
Sir, not visible. So,
my slides are visible.
no, sir. I think
Yes,
it got off.
I will share the new screen. Now,
Yes, sir.
Yes, sir, it
It
is
is
visible.
visible.
okay, I will start with the first itself. Yeah, this is the home page of ensemble plants. From this, we can choose with the glycine max, MYB 100, sorry, MYB 12 transcription factor. This all genome, we need to select also glycine max and then go with the search box. Like we can get the sequence to, we have like we have got the two submission links. We can go to the first one, glyma sequence. Yes, we can get the uh, table like this, like transcription ID, protein, how much the protein amino acid length, and then uniprot ID, and then followed by the graphical illustration. From the graphical illustration, we can get the pink, the dark red color, which highlights as in protein coding regions. From this protein coding regions, this is the protein coding region of the particular segments. We can click this. From this, we can get the transcription ID, gene gene ID, followed by the protein ID, and then GC content of the particular gene, followed by the chromosomal locations. In the particular gene were located in the chromosome number 6, and then position of the length of the sequence from the compared with the around 282,598 base pairs to 282,127 is in start from the stop codon. And also the gene type is in protein coding transcription, and then strand is in positive or the negative strand, and then base pair length 1,530 base pairs, amino acid length, along with the complete informations were present in the uniprot genome sequencing link. From this, we can check with the transcript ID. We can click the transcript ID. Yes, for this, the location along with the transcript ID, the transcript as in one exonic region, followed by the 18 domains, and then 29 oligoprobes. All the information were present in the ensemble link itself. We can click the first exon. Yeah, this is the complete genomic information of the MYB12 of soybean. Yeah. This is we can check with the which segment is coding and then which segment is a non-coding sequence. The we can check with the blue color. The dark blue is in translated sequence. This particular sequence I will highlight, like this sequence were were translated into the protein form, along with the green color signal. The green color sequence were the flanking region of the both forward 5' and then 3' end, and then darker that orange color like UTR, 5' UTR and then 3' UTR were presented. We can copy from the sequence like flanking region like this. Sorry, we can copy from the sequence like UTR sequence 5' UTR and then translated sequence followed by the 3' UTR. Then we can copy the sequence from the site itself. Otherwise, we can directly go with the like what blast analysis. Also, we can perform blast analysis from this website itself. We can check with the blast means we can click the sequence. Yeah, this is the DNA sequence, and then from the glycine max, and then this is a genomic databases like search sensitivity is a normal, and then we can go with the run option. The program was run. Yes, we can get the results. We can view the results. Yeah, see the complete information from NCBI. We can get the graphical illustrations like a pink red color format. From this, we can get the first we got the chromosomal identification followed by the sequence alignment of the blast analysis of the like MYB 100 with various sequencing. All the informations we can get this similar like NCBI format. This is one of the software, and also whether this screen is visible, that new tab.
So we can see
This
the
tab is
results.
working. Visible. This have typed like we can go with the one more software, namely 𝐵ITOGEOM. Yes, thank you, thank you.
Yes.
Yeah, this is the 𝐵ITOGEOM sequence retrieving platform. This platform also so peculiar for the plant species identification only. Yes, in this we can first check with the which species we need to retrieve. First, I will check with the we need to check with the particular plants alone. For best example, I will go with the Solanum lycopersicum. Have selected the Solanum lycopersicum genome. From this, I have to select a single gene itself. I will enter like AUX OX22. OX22 is a gene which is highly responsible for the transport of auxin from the cell to cell contact. This is a polar transport mechanisms, highly transport for the signaling and then transport of the auxin from the cell to cell, because then only the plants were grown enormously. That is the major activity of OX2 gene. Right? We have selected we need to select the first one genome and then OX22. We can go with the search. We need to the OX22 is not present in the particular sequence. That's why we'll select that glycine max. Yes, glycine. We have selected the glycine max. We'll start the first. Wait. OX22. Just a minute. Phytogene. Glycine max. Have selected the glycine max. Then find genes by the keywords. Keywords means just type into gene names. Then we select the search box. can just, we'll select Aragis hypogaea. Then just I'll select ox alone. Go with search. Yes, we can get we got that around maximum 10 similar sequences from the already previously submitted data. From this, we need to check each and every genes. For this, I will select a particular sequence like auxin responsive protein. I will select this. And then we can click the particular sequence. We got the sequencing like complete length of the sequence, then followed by the informations like genomic informations. Like yes, this is genomic sequence informations. Like this is transcriptomic sequence informations. Like this is complete coding sequence. This is protein sequence informations. Whatever, if you want, means we can retrieve the sequence from this. Then one more thing, from this have around only coding sequence alone. Have selected the first sequence. You can go to the browser alone, browser search box. Yes. Yeah, we can visualize the first thing. I will highlight this particular. We can check with these genes like green color is in 5' UTR, blue color is in complete coding sequence, and then red color is in 3' untranslated region, that is called as in 3' UTR. This sequence looks like this. We can check with we can see that 5' UTR. I will highlight. This is 5' UTR regions. First 5' UTR regions, then followed by the coding sequence. This is coding sequence, first highlighted. This is first exon, this first coding region, then followed by this white color background is an exon, intronic region, non-coding region, then followed by the coding regions, then non-coding region. Similarly, back to back coding sequence, non-coding sequence like exonic region and then intronic regions were presented in the genomic sequence. This is the data were stored in the form of DNA. This is a complete 5' UTR. The pink color shows that a 5' UTR sequence. This is the genomic informations. We can check with the transcriptomic informations like this. This is the only the mRNA sequence that also which present in the 5' UTR and then coding sequence, then followed by the 3' UTRs. Like similarly, this complete coding sequence, this is a complete coding sequence of the auxin. Yeah, this is how much we can retrieve the sequence from the phytosome itself. The complete sequence shows around 3.4 KB in nature. And also we can check with the peptide sequence. This is peptide sequence. The dark color peptide, which indicates as an active sites. So, sorry, the complete protein information sites. Along with the activity like expression patterns from the protein, the genes were expressed in various parts like pericarp, fruits, leaves, and also seed, vegetative shoot. All the informations were deposited in the form of graphical illustrations. Like we can check with the particular informations like in the aspect of graphical illustrations, like in which position, then which areas the particular sequence were highly expressed. This is the expression pattern of the spatial this is expression pattern is also known as a spatial temporal expressions. We can retrieve this in how this web platform is a highly useful for the intron exome graphical illustration of the particular genes. I will explain in last software that is called as an exome designer software tools. This particular sequence, we can copy the genomic sequence. We can copy complete sequence. We can copy the sequence and we can store into a new WordPad. We can copy and then paste into the new WordPad. This is how we can retrieve the complete sequence from now apart from the NCBI, like Ensemble plant and also a phytosome software itself. Along with this, we can apart from this, the complete sequence we need to check means like this is a predictory sequence, not is in our designed sequence. For we need to check with our desired targets, means we can amplify the respective host gene. Then only after that we can go with the sequencing analysis. Then we need to compare with the already submitted data and our sequence data. After that, we can check with the blast analysis. The particular blast analysis showed around maximum query length is in 99.99%. If anything less means we can go with one more time sequencing, and then one more time we can repeat the experiments like previous one. This is how we need to check, retrieve the respective sequence from the various platforms like NCBI, Ensemble, and phytosome softwares. Apart from this, I will explain one more software like normal ensembles. Yeah, this is platforms. Basic platform for the ensemble, like we can retrieve the microbial databases, microbial database, and then animal databases also. You can retrieve the basic platforms like NCBI. If you want to check with the specific databases, means you can go to browse, you can read, and then you can check with the particular designed softwares. If already I mentioned like ABRC, if you want to check with the ABRC softwares, means Arabidopsis biological softwares, means we can check with the ABRC websites like Try. That particular platforms which possess only the Arabidopsis sequence informations like that. If you want to check with the soybean sequence, means go with the GLIMA sequencing platform. If you want to check with the tomato, means directly go and check with the tomato databases, like which is hosted by the University of Hyderabad, Telangana. Just a minute, I will share. System is not working. Like Ensemble, Ensemble. Yes, this is the platform. Ensemble Genome Browser. If you want to check with the any kind of specific databases, means you can read previous literatures, then only you can analyze, you can identify the respective genes with the functions. For a best example, if you want to analyze the insulin secreting genes, means if you want to best examples like a particular gene and then particular products, we can know the genomic informations, then only we can design the guide RNAs. Yes, one more questions were arised from the like, yes, CDS sequence. Yes, this is this that the phytosome we take CDS sequence from the design guide. Yes, we can take from the guide RNAs. We can take the CDS from the phytosome itself for the designing of the guide RNA. Not only the ensemble. This is only gene, only the platforms, we can retrieve the sequence, and then we need to confirm the sequence from our designed targets. I'm working with the soybean cultivar X, but already the whole genome sequence, the crop cultivar is some other crop. For each and every crops having dissimilarities, like each and every 0.1% of the inheritance is different. That's why we need to check with the PCR amplification, then go to the sequencing analysis. Then we need to check with the alignment of using mega softwares or otherwise multiple sequence alignments. Then only we can check with the like complete informations like conserved regions and the non-conserved regions, whatever it may be. Like that, we need to check with the blast analysis. It shows around maximum similarity 100%, or otherwise we can repeat. We need to repeat the sequencing results one more time. Yes, from this first sequencing of sequence retrieval and sequence and then retrieval system was completed. This is one of the for most used softwares. Yes, in all softwares we can check with the already submitted sequence like human, mouse, zebrafish, and a number of animal models, and also a number of microbial species also submitted. We can check with this also. And pig, human, mouse, zebrafish, all the informations were available from this. We can retrieve the sequence from this platform itself. But this particular ensemble platform was already linked together with the primary databases like NCBI and UniProt. We can directly retrieve from the sequence from NCBI or UniProt itself. Now my slides are visible. Whether my slides are visible? Yes, thank you. Yes, like this is a home page of NCBI. We can type the gene name and followed by the go with the search options. Then we can retrieve the sequence from directly from the NCBI itself. Or otherwise, we can directly go to the ensemble plants. If you want to, if you are those who are working in plants, we can retrieve the sequence from ensemble plants also. This is a home platform. Like this is the we need to type the gene name like ox, whatever it may be, gene name or the complete sequence like ox means auxin responsive genes. We need to type and then search into the website, and then we can get the results in the form of description, genome ID, species, and then locations. We can directly go click with the particular gene ID. We can retrieve the a sequence in the form of either 5' UTR, 3' UTR, and then followed by the complete coding sequence along with the genomic information from starting point to the start point, and then length also. This is retrieval system from the ensemble plants. This is phytosome. Phytosome is peculiar for the plants itself. We need to click the particular respective species and then check with the genome of our interest. Then we can type into the sequence name and then select with the particular species. We can check with the complete gene, select the particular gene. We need to get there around more than 10 to 15 sequence, means we need to check with each and every databases. If you want to check, go with the precise one, means we can select the particular genomic informations. Then we can get the informations like this, genomic informations like 5' UTR, 3' UTR, then followed by the CDS. This is the information of the genomic sequence. After that, we can check with the transcriptomes. The complete exomic regions, intronic regions were removed from the transcriptomic data. followed by the, if you want to check with the complete CDS alone, the coding sequence alone means we can check with the CDS retrieval systems. If you want to, I'm working in, I don't want any genomic information, sir. If I want to check with protein alone means we can check with the peptide sequence. We can retrieve the peptide sequence from the databases itself. After that, we can check with the expression pattern in which gene were expressed in the seed, seed, leaf, stigma, pollen, then style, whatever it may be from the leaf, root. If you want to check with the expression pattern, the particular informations were already deposited in the format of graphical illustration. We can retrieve the particular data itself from the phytosome platform itself. Yeah, if you want to check with the blast results, we can directly go to the blast analysis also, similar towards the previous one, like NCBI blast results also. We can this platform, this results were showed that blast results, then followed by the e-value, then score matrix analysis. If you want to check with the pairwise means the particular results were visualized in the format of pairwise. If you want to check with the local alignment, pairwise alignment means we can visualize whatever it may be. We can get the we can export the data in the format of PDF or in the format of HTML file. Yes, I think this is a core critical topic, like guide RNA design and guide RNA design for the targeted selected target. For this, we have plenty of softwares. The first one, ChopChop. The second one, CRISPR scan. Third one, CRISPR P2, CRISPR Direct. Fourth one is CRISPR Direct, and then finally CC Top. From this, I will explain 2 or 3 softwares, like in the PPT itself. Tomorrow we'll start the session with the hands-on with the guide RNA designing, followed by the sequence similarity analysis. Yes, this is home page of ChopChop. From this, we can check with the otherwise we can directly go with the ChopChop. Then we'll wind up the if you want to have any queries, we'll discuss. After that, we'll wind up. Yes. First, I will stop the sharing. I will go with the screen sharing. Share. Yes, whether my slide, my screen are visible?
Yes, sir.
Yes, thank you. Yeah, you can directly type with the ChopChop. Sorry. Google. From the Google itself, we can directly go with the ChopChop. Yeah, this is the ChopChop platform. It's a freely available software, mainly used for the guide RNA designing. Yeah. Already we have selected the sequence, like we have copied from the sequence from the notepad. ChopChop. From this, we need to select the species. This is the sequence from Arabidopsis thaliana. All the plants, animals, microbes, bacterium, all the databases were linked together in the format of ChopChop. We can directly check with that also. From this Arabidopsis thaliana, I have selected the Arabidopsis thaliana, and then we need to check. Already I have pasted, copy the sequence. I need to paste the sequence, then using the CRISPR-Cas9, and then knockout format. If you want to go with any kind of specific like Cas9 IKs, and then CPS1 or Cas12 or Cas6 or Cas13, or then talents, if you want to check with the particular thing, and then if you want to go to the knock-in, knockout, activation, repression, and then nanopore enrichment. If you want to check with, we can find out the guide RNAs itself. Already we have pasted the sequence in Arabidopsis thaliana. Then we'll go with the target identification for the efficient knockout of the particular target. Yeah, the software is running. Yes, if you want, it will take more than 10 to 15 minutes at the time. Nanopore enrichment is also one of a kind of CRISPR technology to check with the efficient knockout using NGS methodologies. Yes, we can discuss because this particular software will shows around it will take more than 10 to 15 minutes at the time. We'll discuss like anything is there means we can discuss. If you have any queries, you can ask, please.
Sir, till now you were teaching how the information about any part of a given genome, required genome, can be found by using the already published sequences of various genomes of various species. Is that correct, sir?
Can you no? Can you repeat the questions because your voice is very low, please?
But till you came to the 𝐄𝐃𝐈𝐓 software, you were
Yes.
teaching how we can get the information regarding the genes anywhere or any part of the genome of the required species, on which we will then do the gene editing. Is that correct, sir? Have I understood correctly what you were teaching?
Yes, correct. If you want to check with any kind of gene, yeah, you can use because you have a laptop, already I have submitted the sequence, but the software is currently running. If you want to go with same software, same methodologies, you can check with the in your system. Initial. Okay. First, we need to, for efficient knockout or knock-in, we need to check with the target first. If you fix the target is must. We need to check with the previous literatures, then only we can find out the any kind of genes. Like if you want to knock out a particular gene, the particular activity of the gene should be known. Correct. What is the major glycolysis genes means already the particular genes informations were already sequenced, submitted, and then particular informations were well characterized because glucose to glucose 6-phosphate 𝐂𝐑𝐈𝐒. This particular gene was already sequenced, submitted. If have any defected from the yes, if you want to retrieve the sequence from whatever it may be, so like we can retrieve the sequence from NCBI. If you want to check with the peculiar sequence means go with the particular platforms like ensemble basic platforms are available like plants, microbes, animals. If you want to check with the plants means ensemble plants. If you want to check with microbes and then animals means go with the ensembled. Phytogome also available. If you want to check with particular precise means we can go with the precise sequence, then retrieve the sequence from the databases, and then you can go with the target uh, guide RNA designing tools. Before going to guide RNA designing, we need to check with the in our sequence. Like I have a particular cultivar. Particular cultivar means I have one cultivar X. The name is called as an X, but already the submitted sequence is called as an Y. But there is a huge differences between X, Y. That's why we need to check with the amplification using the PCR. Then we can go to the Sanger sequencing, or otherwise we can go to the NGS sequencing depending upon the amount which you have. Basically, we can go to the Sanger sequencing, or in the case of means like a primer walking technology also majorly used to predict the maximum query coverage. We can check the sequence. After that, we can go to the chop and guide RNA design tools. The first one is a chop. Yes, exactly. We got the results like this. This is the color-based identification technologies. We can check, yeah. I will scroll down. We got around 140 guide RNAs for the MYB12 transcription factor. Okay, this is based on the colors like green color, yellow color, and red color in nature. Green means highly efficient guide RNAs, followed by the yellow is in moderate, and then red means as in poorly working guide RNA sequencing results. Yes, this is rank-based screening. We have to select the first rank. We can check with the first rank where it is located. The first rank which is located in the in between the sequence number 1 to 200 base pairs. Yeah, we can go with the we need to select the sequence, the guide RNA sequence. We have selected the first sequence. This is the target gene, the target sequence that is end with the NGG. That is NGG, the final sequence that is called as an NGG. We can check with the NGG, that the right hand side and then left hand side. Left hand means 5 prime. The right hand side is in 3 prime. The 3 prime end with the NGG, CGG. This is called as a PAM site, and then complete genomic locations, the sequencing location, and then stand, which is whether it's in positive strand or the negative strand, and then percentage of the GC content, and then self-complementary, and then mismatch, and then efficiency. This is how the rank-based screening of the designed guide RNAs. Here, the mismatch should be completely zero. Correct. The self-complementary also should be zero, and also maximum we can choose with the positive strand. Here we can check with the mismatch MM0, MM1, MM2, MM3. Here we can all the columns, all the MM should be the 0, or otherwise the self-complementary also here also 0, but the poor yes, only one sequence which shows the 0, 0. Yeah, we can check the guide RNA number 47. It shows the maximum mismatch 0, or otherwise the off-target effect also should be high. Mismatch is directly proportional to the off-target. Off-target means nothing but the particular guide RNA will go and binds the unwanted positions, unwanted targets, and then the particular guide RNA will cleave the entire genomic information. Thereby the irrelevant mismatch, irrelevant protein productions where occurs. For that purpose, we need to, the mismatch should be the 0. The mismatch is directly proportional to the off-target efficiency. Off-target should be 0, and then mismatch should be the 0, and then self-complementary also 0. But here the rank number 47 shows that self-complementary one, the GC content is very poor, and then stand number is minus. In this time, we can check with the multiple guide RNAs, like guide RNA rank number 1, 2, 3, 4, 5, 6. Don't stick on only one guide RNAs. You can check with the n number of guide RNAs from the single target itself. Yes, we can click the sequence. Whether my slides are visible?
Yes, sir.
Not in slides. I am working in ChapChap online tool. Okay, we have selected the guide RNA number 47. Yeah, it is look like this. This is the complete target sequence, like end with the NGG, NGG. Yeah, the particular cut will be the pale blue color, the arrow mark dark in black color, and then the pale blue color is in cut position from the target. Here we can check with the after complete editing process, we can check with the editing process through PCR analysis. Like this is the first fifth PCR set of primers. This is 4, 3, 2, 1. for using this particular guide RNAs, we can select the primer sequence from this here, first primer target, and then finally the complete sequence, the complete product will amplified. After that, we'll go with the sequencing analysis. Then we'll check with the control, and then followed by the edited lines where the indel mutations were occur, and then easily we can identify the editing indel mutation screening. For this approach, based on the rank-based designing of the guide RNAs, followed by the easiest prediction of the guide RNAs through the PCR sequencing analysis. We can check with the PCR analysis, then the product size also we can get, and then finally we can check with the both the forward primer and then reverse primer using amplification of the respective PCR. Yes, we can check with the off-target efficiency. We can go to the location, and then mismatch should be the 0, and then sequence also. This is the sequence should be the should be nature of the 0. For this, we can use for the guide RNA designed guide RNA. We can copy from the sequence, and then this is the forward sequence, and then we can design the reverse of the reverse guide RNAs. Like, but one thing is that, but here the guide RNAs GC content is very, very low. That's why we can move to the next guide RNAs. Like which one isn't shows the maximum GC content. Like we can check with the guide RNAs. Here we can click with the guide RNA number 6, but here also self-complementary one. But it shows that the maximum, yeah, sequence. We can check with the mismatch like this, and then chromosomal location mismatch should be the 0, and then product length, and then PCR primers, and then also the cut position apart from the NGG. This is, we can check with the NGG. See that the NGG, the CGG, like see that the NGG, TGG. This is the guide RNA TAM site. After that cut should be the year. This is the randomized cut. We can check with the using PCR sequencing analysis only. This is how we need to select the guide RNAs from the 𝐶𝐵 website itself. Yes, have any doubts using 𝐶𝐵? If you want to check with any kind of sequence like plant, microbe, animal, you can retrieve the sequence from the NCBI and then directly go to the 𝐶𝐵 from the web tool itself. This is freely available software. You can retrieve the respective guide RNAs from the website itself. After that, we can go to the synthesis process of the respective guide RNAs. Then we can annealed using respective temperatures. I will discuss with later. Then we can cloned with the normal restriction digestion and then ligation method of cloning process. After that, we can mobilize with the E. coli transformation. Then finally go to the respective downstream analysis like PCR screenings, whatever it may be, like RT-PCR analysis like that. Have any doubts?
Or will you discuss the DNA extraction also when after teaching us how to synthesize the guide RNA? Will you teach us the extraction, DNA extraction also, sir?
I couldn't able to understand, sir. Please.
Sir, will you discuss DNA extraction process also?
DNA
extraction.
Yes, DNA extraction.
Will you discuss that also, sir, in future classes?
During the class, yes, I will discuss, because after this is a basic process. First, we need to design the guide RNAs. Then we'll go to the transformation. After that, we need to go to the downstream screening. Then only at the time I will discuss with the plasmid DNA isolation, then RNA isolation, then protein isolation, followed by the some other isolation protocols for the efficient knockout identification process. Later, we will discuss the isolation of DNA, plasmid, isolation protocols. Yes, sir. Yes.
Oh, thank you, sir. Thank you.
Yes, thank you. Thank you. This is a 𝐶𝐃𝐈𝐒𝐏. If you want to check with the software, means you can analyze using a number of genes are already submitted, like well-known genes. Yes, efficiency is must important. If the sequence like it shows that around 70% means we can click with the first rank. That's why this isn't based on the rank based. If the rank is directly proportional to the efficiency of the particular guide RNA, the efficiency is based on the rank. Here the 3 ranks based on the color and followed by the efficiencies. Already we have discussed, like first green color is a complete efficient targets, efficient guide RNAs, followed by the moderate guide RNAs, and then low rank guide RNAs like in red in nature. And then also the particular guide RNA showed that rank number 1, position number 138, like in which position the guide RNA goes and bind. All the informations were presented in the particular software itself. We can retrieve the, we can copy the respective sequence and reverse complementary of the reverse. We can synthesize from the respective company, and then we can directly go to the cloning process. Any other questions need to be discussed? Yes, obviously, but one thing, all the efficiency is not like that. If you want to check with the mismatch, all the mismatch should be the 0, self-complementary 0, and the efficiency should be I. Then only we can select with the particular guide RNAs, or otherwise the complete experiment will goes to fails. For this, avoiding this critical conditions, we can go to the multiple guide RNA synthesis. We can select the multiple guide RNAs from, yes, I will, yeah, this is 5 prime, this is a 5 prime end, this is the 3 prime end you can consider, and then this is in between protein coding sequence. The particular sequence should be the 5 prime or 3 prime, because the 5 prime 3 prime is the most essential segments for the proteins. If the cut is 5 prime, means the complete informations of the protein will collapse. After that, the CRISPR experiments will go into the success. We can efficiency is directly proportional to the rank. Yes, I will discuss with my senior. They will definitely forward the yesterday's recordings also in the respective link, which already provided in the WhatsApp groups. Have any queries? Shall we wind up? Today we'll learn about how to retrieve the sequence from various platforms, then followed by the guide RNA designing using 𝐶𝐃𝐈𝐒𝐏. Tomorrow we'll discuss about CRISPR-P2, CC-top, followed by the CRISPR direct, and then eCRISPR for the designing of the efficient guide RNAs. Yes, thank you. If the mismatch is yes, if the mismatch is very high, means we can go to the some other targets, like rank 1 is a highly mismatch, means we can go to the second rank. That is not at all, this is not problematic one. We can check with the 1, 2, 3, 4, 5, 6 based on the requirement only. Don't stick only one guide RNAs. You can select multiple guide RNAs, and then you can synthesize, and then you can clone into the respective vectors, and then you can immediately transform to the organisms. This is based on 1 to 5 days or 1 week to 14 days of experiments only. You can, if you have a very good transformation protocol of your respective host, definitely the particular guide RNAs will works very efficiently. Yes. Thank you. Thank you.
Sorry, just a last one question, sir.
Yes.
But is the cloning of the guide RNA done by the replication of the vector? Is that how it is cloned? Is that how the guide RNA is cloned?
Yeah, that is based on the normal cloning process, restriction digestion process. If you want, if already we have designed the guide RNAs, we got from the primer designing companies with the respective vials. After that, we need to dilute the respective oligos. Then we need to anneal into the 95 degrees Celsius from the 95 degrees Celsius for 30 minutes, and then gradual reduction of the temperature should be the 25 degrees Celsius. After that, the respective oligos should be digested using respective restriction enzymes. Then followed by the we can directly clone into the respective vectors like whatever it may be. If you want to go to the plant vectors, you can use with the PHSC401, or otherwise you can go with the PREGB32. This is PREGB32, and then 33 vectors are the normal vectors from the plants, animals, and then bacterial itself. You can clone into the respective vector and then directly go to the transformation protocols.
Okay, so thank you. I will
Yes.
study it myself.
Thank you,
also.
thank you, sir.
very
Thank
much.
you, thank you. Yes. Have any queries? So chat box have some queries. Sure, I will discuss with my seniors and then I will let you know the changing of the times. If you go to the NCBI software, you can check with the human genome sequence for your respective experiments. If you want to knock out the particular human gene, you can retrieve the sequence from the NCBI and then you can go to the experiment predictions. Yes, bioinformatics is in core. Then only we can design the guidance. Otherwise, I will just a minute. I will give the one sequence. I will give. Can you design the guidance sequence? It is not possible. Correct. For this purpose, only we need to rely on the bioinformatic tools for the designing of the guidance. This is highly interconnected towards the designing of guidance and then sequencing informations. Like we got the sequence, we amplified the respective product, and then we gave the sequencing analysis like either NGS or either Sanger sequencing, either primer working methodology. We need to analyze the respective sequence, means we have to rely on only in bioinformatic analysis. For this purpose, only bioinformatics is main core subject. We're linked with the biotechnology. Biotechnology means all the kind of technologies like microbial, animal, plant, whatever it may be. Yes. Yes, but I don't know about the exact human genome in NCBI. You can check with the previous literatures. That's why you can refer n number of articles. Then only you can understand what is the major genes, which is the major function. We can check with that also. I will. Yeah, if you have any doubts, you can check. You can let you know tomorrow. Tomorrow also we'll discuss what is the if I have select the gRNA with the least mismatch or the highest efficiency. Yeah, least mismatch with highest efficiency. You can. This is bioinformatic tools, correct? We don't rely on this one. This is one of the software. You can go with some other softwares. I will discuss with later. Don't stick on only one software, but don't stick on only one guide harness. You can synthesize n number of guide harness. You can use n number of basic platforms. You can design the n number of guide harness, and then you can work. Then only you can succeed in your experiments. Then only you can conclude your whatever it may be, thesis or research, whatever it may be. You can no need to rely on the only one softwares. Yes. Yes. Thank you. We'll discuss on tomorrow also. Thank you. Thank you. Yeah, this is a trial and error mechanism. First, you can go to the trial and error mechanisms, and then you can go to this is basic bioinformatic analysis. Then you can go to the respective analysis. Then only you can understand the particular process. No. We can go to the only vectors which is highly efficient for the transformation purpose itself. For plants means PBR 320. Sorry. N number of binary vectors are also available. You can choose the respective vectors, then only you can clone into the expression vectors, not in your replication vector. Expression vectors also one of the option. Both the vectors were replicated into the E. coli and also the host of the target host. We can choose the respective vector and then respective guide RNAs. Then only we can succeed in our experiments, or otherwise we can't. Yes, thank you. If you have any queries, we'll discuss on later. Thank you. Yes.